Yeah, everyone, we're going to have breaks after this one. This is our last talk, and then we'll be able to engage with each other during the break and work on questions. So our next speaker is Shaun Jackson, who is a pediatric nephrologist and rheumatologist from the University of Seattle in Washington.
Yeah, so thanks. So I must acknowledge that I was originally asked to give the CAR T cell talk, and I was the one that said, do you mind if I give bispecifics? I know a little bit more about that. So, in reference to my colleagues, Brad told me the goal of this debate was not to win, but I'm going to show one piece of sort of animal data right at the end, which will hopefully emphasize why I think bispecifics are going to be the way that we will go in the long term. So I have some disclosures. So just to sort of frame, I think the most important thing is to talk about what bispecifics are and how to think about them in the spectrum of monoclonal antibodies versus CAR T cells. So every time I re-upload this figure, it goes fuzzy again. But you can see on the left here, this is the obinutuzumab, just a monoclonal antibody binding CD20. This is a CAR T cell where you generate a T cell which is going to target that target. Bispecifics are really in between. It's a protein therapeutic, which is a T cell engager. It brings CD3 on the it binds to its target, whatever that is, CD19, CD20, BCMA, and then engages CD3 on the T cell side to induce killing by endogenous T cells. The most widely studied drugs, so this is a general schema of what happens. Once, if you had two antibodies, they wouldn't do anything. If you combine them, they will bring that T cell synapse together, cause T cell cytokine release and induction of killing mechanisms, and in this case, kill the target. But of course, just like CAR T cells, a B cell leukemia, lymphoma therapy becomes a B cell targeted therapy. And I want to sort of highlight one thing about how well this drug actually works and how it's changing care. So this is not autoimmunity. This is pediatric leukemia. And I mentioned this because it's a study that came out a couple of months ago in the New England Journal, and one of my colleagues was the senior author on this study. Now, what people may not realize is that the survival rate, and this is not survival, this is disease-free, relapse-free survival. The actual survival rate is higher than this, of chemotherapy in standard risk acute lymphoblastic leukemia. The survival rate is actually really good. About 85% of patients respond with standard therapy. And if I was wanting to get involved in a trial, I would not want to get involved in a trial where everybody does that well. But in this case, they added blinatumomab, as they've done with all these leukemia trials, and just had this remarkable response. There was an extra 10% 3-year relapse-free survival with blinatumomab. And I can say that the rates of CRS with this drug were actually very low. It was well tolerated in the context of getting standard chemotherapy, right? So it's sort of been mentioned. So what are the advantages? Well, the obvious advantage is it's off the shelf, and although the drug itself will be expensive when they're first approved, the actual manufacturing is going to be cheaper. It's a protein therapeutic, same manufacturing costs as any antibody. You don't need any lymphodepleting chemotherapy. In fact, you can't have any lymphodepleting chemotherapy because then you will ablate the T cells that are the killers. And it has the theoretical potential for CAR T cell-like degree of B cell depletion. What are the disadvantages? So we've mentioned the risk of CRS and ICANS, and that's sort of a predictable risk. This is a drug that's in the interface between a monoclonal and a CAR T cell. It's causing T cell activation, and so it would be expected that you would get some level of CRS. However, I anticipate that this will be likely low in autoimmunity for the simple reason that CRS rates in the CAR T cell population are lower in autoimmunity compared to oncology. You've got a normal B cell count, not a sort of body full of kilograms of tumor, etc. Some of the current drugs in the original BiTE format lack an Fc receptor, so their half-life is very short, has to be given by continuous infusion. So those patients in the leukemia trial, they go home wearing a little backpack which infuses the drug for a month. It's kind of incredible that that's how it's done. But there are new forms that are generating Fc receptors. It's a protein therapeutic, and so it has the theoretical risk of immunogenicity. Blinatumomab has very low immunogenicity rates. But if you look at some of the newer agents that have been developed for oncology, actually a lot of the programs have failed because of the immunogenicity of the product. And I sort of mentioned this to sort of say, like, this is a spectrum of the current ways in which protein design people are thinking about putting antibodies together. And this is a small subset. There's IgM-based forms. There's parts that they put the CD3 on one end, 2 different T cell targets. I think actually once you get more and more into these Franken molecules, you're going to have more immunogenicity. But maybe that's not a problem in autoimmunity if the goal is just to, get short-term depletion of whatever target we want to aim. So the question then is, do we think that BiTEs are actually going to achieve CAR T cell-like effects in terms of B cell depletion? And I should say that there will be oncologists in the room who will say, we have had patients who failed blinatumomab and then had a successful response to CD19 CAR. They're not the same, right? So will we get CAR T cell-like B cell depletion? And that's very hard to prove. Like, we got to wait until we generate that data. We don't have the access to just go into and biopsy every single lymph node, spleen, kidney, etc. And so we're doing the obvious thing, which is trying to model this in animals. And so what we did, after a lot of tweaking with the design, is we made a mouse version of blinatumomab. So it targets mouse CD19, mouse CD3. It's never going to go into a human patient. It's not going to, we're never going to make any money out of this, but we sort of want to use this as a way to test whether this drug will work. And this is the results, right? So you can chop up a mouse and you can look at all the organs. It's really easy to deplete in the peripheral blood, but in this mouse, we could deplete in the spleen, we could deplete in the lymph nodes, we can deplete everywhere. If you're super astute, you might say, well, actually, there's 0.05% B cells in the spleen after, and this was just 3 days of injection in this mice. It was a very junior research scientist that did this experiment a few months ago, and while he was sucking the mice, he somehow forgot to take the spleen on one of our animals. I don't know how you do that, but he just forgot to do it. But he'd already set up his flow panel. He was very worried about lacking something in how he set up his design. And so he ran PBS in one of his samples. And what was interesting is you've got 0.05% B cells, just from B cells stuck to the tube. So I think that this is very deep B cell depletion and could be the solution that sort of gets there if we think CD19 is the target or BCMA, whatever. And that's it.